Can I have the questions left from session 3 yesterday? We have to answer those before we move to our panel. What are the challenges for post-marketing genomic data collection? I'm afraid we have only one person in the room to really answer those. Our Pharmacogenomics Working Party and PRAC committee member, Kunying Yue. Milady, can I ask you to answer? Questions are displayed on the screen. What are the challenges for post-marketing genomic data collection? How spontaneous ADR reports related to possible genetic polyformisms may trigger pharmacogenetic research? That's the first one.
Yes, this is the question was put on the screen from yesterday. Well, I'm sure that people from industry and people from regulatory authority and academy will all feel these challenges. While the collection of blood samples in clinical trials is relatively easy and has been a routine, there's no particular requirement for the post-marketing setting in the systematic collection of spontaneous reporting related blood sample collection. There have been some initiatives in different countries or different parts of the world. There are Eugene projects. There are psych project, international psych project in the US. And I think these consortium involve both industry and regulatory authorities. I think one of the challenges is the privacy or data protection. Some of the stakeholders may not have all the information on the patients and may not have a permission from the physicians to get a hold of the blood samples or genetic materials. There have been some publications, I think, from the Netherlands, the role of a regional center in helping physicians to perform genetic tests. If they identify some signals from spontaneous reports where the adverse reaction probably related to drugs with the polymorphisms, they would encourage the physician to perform genomic tests and increase the knowledge. The question is how to systematic collect genomic samples for new drugs where we do not know very much about the safety profile, about particularly the serious adverse reactions, either idiosyncratic, we don't know the mechanism, that have been discovered late in drug development or post-marketing, or there is mechanism pharmacologically related, or there is mechanism for polymorphisms, but we would like to know the magnitude of the problem at the post-marketing setting. So I would like to hear the opinions from different experts here and stakeholders. What's your opinion?
Please.
Yes, hi, this is Eric Lai from Takeda. First of all, I just want to point out that even for clinical trials that are phase 2 and phase 3, collection of samples are really spotty. And even though you might think that it's recommended in all of the regulatory guidelines by FDA and EMA, just because you recommend it, it doesn't mean that it is done consistently. So that's an illusion. Okay, if you think that that is reality, even for the biggest company that are really in the pharmacogenomic arena, like Roche and Pfizer, if you talk to those folks, it is not consistent. Oncology, absolutely, but any other therapeutic area, that is not the case. And as far as the post-marketing is concerned, I think it's one of the biggest hole as far as safety is concerned. I'm a strong believer that there are a lot of techniques that you can use to detect the signal on a genetic basis for safety. However, without collecting the sample is useless. And once you have reported the signal by whatever analysis, meta-analysis or publication by a researcher, it is almost impossible to go back and collect the samples because now you're talking to the lawyers, not the physicians and the patients. I know this because I was the one who did the Abacavir 5701 when I was at GSK, and that was a painful experience in order to go back and collect all of those samples. So I think that if this is really something that all of us want to seriously look into, it has to have a regulatory framework by both FDA and EMA to really provide exactly how to do this. You don't want to end up with 10 companies doing 20 different things of collecting post-marketing and try to analyze it differently. That's not what you want to be. And I think that we have an opportunity here to really set up properly to move forward.
Thank you very much. Any other comment on this question? Yes, please, Bruno.
No, just to confirm that there have been many initiatives over the last 10 years or so, probably, with Delhi, Uragine, and so on, of trying to collect DNA samples from those who actually got the adverse events and compare them to the other groups. It's a tremendous task, and the success rate, I mean, compared to the efforts that have been injected, is extremely small for the time being. So I don't know, perhaps the NCEP initiative in Europe is going, some people hope it's going to make a change in this regard, but I think I agree with you, we need some strong incentive and input into the system to make it change.
I also started the Severe Adverse Reaction Consortium after the SNP consortium with Arthur Houghton. It was a really difficult thing to do, and try to get all the farmers together, and then try to get the farmers to donate samples for the consortiums. It is not a difficult thing, and at the end, because the methods of collections were very different, a lot of those efforts was really wasted. We don't set it out properly in the beginning of a framework of how to collect this, how to get the sample. It's not going to get done.
Interesting discussion. We need to move to question 2, comparison with DNA from patients without the safety efficacy concern. Can I ask again, Kunying Yue, milady, please?
I think these two questions are related. If you just have a sample from cases, it doesn't tell you so much on the magnitude or the real risk. You need to compare with someone with the controls, and the idea is that try to have sort of a systematic collection of data. You have samples data from both cases and the controls. There are several approaches mentioned here. We have a biobanking, the consortiums mentioned, and NCEP is mentioned here, but how should this be organized? What is the best design for what kind of safety concerns? And here again, I would like to hear your opinion.
Marisa Papalouko has a comment to make before we move on.
Just with a disclaimer, with this not the view of the agency and just a personal reflection. I think that there are elements in what has been just said so far which would prepare for a more, I would say, organized work to be done, at least in Europe, in order to address these questions. Post-marketing genomic data collection. We heard from the shade of grays which are around this area of science that there is a lot of learning which should happen after the marketing authorization is granted. So maybe we have to see the things as a continuum and think of NCEP and other clinical network to work together to define the phenotype, to define what we need to have from the clinical standpoint in order to have a meaningful set of data collected at a given point in time. But I think what is really missing, and I think maybe this links to the subsequent set of questions from the panel, is that there is no real an infrastructure harmonized whereby companies and clinical centers work according to a predefined protocol, a predefined set of platforms, which then are put to the service of the community. It's not for the industry, it's not just for the investigator, it's just to the service of the community, and can be used equally for clinical trials post-approval, for example, if we were to think about adaptive licensing and acquiring randomized controlled trial information following the authorization, or for post-marketing considerations and correlation with the adverse reactions emerging. So I think there is an element there, and I would take inspiration from the two speakers we had today, how important it is that, and from the Frederick Nowak example yesterday, how important it is to really establish a framework which then can be improved and harmonized and put on the same pace across Europe. This is really, I think, missing at the moment.
Thank you, Marisa. Any comment from the floor, please?
Yes, Cynthia Sung from Health Sciences Authority in Singapore. I thought I would just share a little bit about what we're doing in Singapore related to the first question. So I think there's less inhibition in Singapore about reporting reporting the race of the ADR report. So in Singapore, there are three major ethnic groups: Chinese, Malay, and Indian. And so one thing that we have done is to look at the proportion of ADRs that come in by ethnic group and see if that is out of sync with the background racial proportion. And in 2004, actually, we did notice that the Malays had more 1/4 of the carbamazepine-induced SJS, even though they only comprise 13% of the population. And then the next month, we saw the report from Taiwan that HLA-B1502 report, and we have now seen that HLA-B1502 is twice as common in the Malays than in the Chinese. And so we have been monitoring, we have found a few other examples. Now, we don't have the DNA to go with those, but we do see signals, and that's maybe one way that we can possibly pick up or generate hypotheses for using race as maybe an imperfect surrogate for some genetic disparity or genetic polymorphism. Just wanted to share that.
Thank you very much. The gentleman who is sharing my haircut, please.
It's Steve Spielberg from FDA. One model won't work for everything, but there are a number of interesting models that I think we should look at and consider. One is a model at Vanderbilt where every patient who comes into the institution and has blood drawn has the option of opting out of having DNA collected and anonymized. This is all done anonymously. It's not patient-centric. It's done with a 23-digit code that is coded for the blood sample, and then followed up, both for disease state as well as for outcomes of therapeutics. The opt-in rate, by the way, is greater than 95%. That has been the case both for adults as well as children. And all the samples collected in a standardized way and analyzed in a standard way. Another approach, the ITCH Consortium, looking at drug-induced severe skin outcomes, where the critical features have been international agreement on phenotype, so that it's quite clear what a definition of Stevens-Johnson, toxic epidermal necrolysis, hives, and other various types of skin conditions are. That has led to the HLA findings, but we're now reanalyzing those data on carbamazepine, doing whole genome sequencing at the National Center for Toxicology Research, which is part of FDA, collaboratively with our colleagues in Liverpool, and we'll see if that model is useful. Third model, again, is disease-specific, and this is related to rapid approval of drugs, where we are approving compounds on the basis of 200 human exposures, and we want to see what happens post-market, of developing cohorts and registries of patients well-studied with agreed-upon protocols clinically, and I can't emphasize the importance of the clinical enough because we get the clinical phenotype wrong half the time, or maybe more, and have DNA collected in a systematic, standardized way. And then we look at these various different ways of approaching small and large populations and selecting those that work.
Thank you. Francois, please.
François here from Ordis. A very short question, a practical one, but as a patient, how much of my body should I biobank? And if I am interested in pharmacogenomics to be tested, if I am for comparison, and by the time this research is undergone, all my tissues have been used for other research purposes, and there is nothing left for this pharmacogenomic study. How, as a patient, can I control the future use of the body tissues and samples that I would have consent to give to research? I think that is also a concern, a practical one that we may have to work on.
Thank you, Francois. Anybody wants to reply to this? Hans-Georg, please.
I'm hearing from industry and academia that the greatest obstacle to this research is exactly what you just described, that you would have to go back to ask patients to consent, etc. My question, I'm not talking as an employee of this organization here, as a citizen, is, do I want to have control over this? Isn't research a generational contract? We all benefit from the research that has been conducted on patient generations before us. I have no interest in controlling, as long as it's anonymized, what is going to happen to my samples.
Please.
One other thing that we should also keep in mind, I'm speaking here as a researcher, is that we're talking about ADR. ADR can be very heterogeneous in definition as a phenotype. So if we are to combine a phenotypic data, clinical data, to perform genetic analysis, one should be very cautious to have homogeneous phenotypic data to be able to get some sound genotypic data from the other hand. So in order to get some pharmacogenetic research on ADRs, and also I agree with Jean when she said that the questions number 2 and number 1 are together, we should ensure that whatever phenotypic data are recorded are very thorough, and very detailed, so that we can actually cluster them together and combine them with the genotypic analysis when it's performed.
Can I ask Inge to display the next slide? The panel members to introduce themselves and to make one comment. Please go ahead, then we have the coffee break.
Markku Pasanen from the University of Eastern Finland and a Scientific Advice Safety Working Party member. And, or should everybody introduce themselves?
My name is Richard West. I'm a patient of Behçet syndrome. I'm also a volunteer representative with Eurordis, which is the rare diseases in Europe.
My name is Julia Stingel. I'm a professor of pharmacology and now working at the German Federal Institute of Drugs. I'm building up a research department on pharmacogenomics there, hopefully.
My name is Ingolf Gaskorbi. I'm head of pharmacology in Kiel in Germany, and I represent the EACPT in the Healthcare Professional Working Group.
Hi, my name's Nick Mead. I'm here representing the European Genetic Alliances Network, and I'm an ex-member of the Committee for Advanced Therapies.
Inge, please display the next slide. This is the question we are going to answer. Panel members, please.
May I initiate one additional question? No.
If it's very, very, very short.
I like to discuss or make an additional to the question which Bruno highlighted to Robert Diaso about these non-clinical models. I made this previously in July, but I don't get any answer. So installation in these non-clinical models, the cells to mice was used as, I guess, subcutaneous. Why not using orthotopological installation of tumor cells to predict more precisely the clinical problem? Now I can.
I'd like to make a comment from a patient point of view. I looked at some of the information that came before the event was to be held, and one that interested me was the reaction with azathioprine for a TPMT. And the reason I was particularly interested is because I've been taking the drug for the last 14 years, and I've taken over 20,000 azathioprine tablets. So I thought my first point of call was I would look at, well, what does my patient information package leaflet say about the drug? And I'd like to just read it to you, just one little snippet. It says here, before you take azathioprine, take special care if you have a condition where you have a deficiency of TPMT. And the important point is, it says, tell your doctor before you start to take this medication. Well, how on earth is any patient going to know if you've got this unless you've had the test? So what I did next was I went and had a look at the EMA website to see what information they gave about azathioprine. There was nothing, because it's not a centrally authorized drug. So I went to the FDA site, and they had a really succinct, full of information, really good. If you were a pharmacist, if you were a clinician, if you were a patient, it was excellent. And it talked about the testing that you should have before you take azathioprine. And it said that they recommended that consideration should be given to having the test. The essence of this testing for azathioprine is that in a white population, you've got a 1 in 300 chance of having a life-threatening disease if you take the drug. Now, when I looked at, the next thing I looked at was the summary of product characteristics, and it talks about rare individuals with TPMT. Now, I don't class 1 in 300 as being a rare individual, because a rare individual for rare diseases is 1 in 2,000. So then I looked at, well, what does the MHRA say? And it was very interesting because they have about 16 different leaflets for azathioprine and urine. And what they go on to say, and as I looked, on their patient information leaflet, there is nothing, no mention, if you look at 25 milligrams and 50 milligram tablets, concerning testing before you actually go on to the drug. So if you're a patient and you're trying to find information about, well, should you go on this drug, what's the implications? It's an absolute maze. So from my point of view, I would like a consistent approach. I would like something that is the same information that you get wherever you are in the world, whether it be Europe and America, and you have sufficient information that you can actually make a judgment on whether you should take the drug or whether you shouldn't take the drug. So I think the thing I would come back to is the question here is, well, what is the testing? Should it be done? And my point is, Hans Jordan made the comment, is the test being conducted? I think probably in the UK it's not. And what happens is the clinicians, after you start taking the drug, monitor you weekly. And if they see adverse reactions, they stop you on the drug, rather than doing the test up front. So my feeling is, from a patient point of view, is that there isn't sufficient information. What information there is out there is difficult to access, and it's difficult to understand. Thank you.
Julia Stingel, please.
Okay. When I read this, it always comes up to me that we should not pick out pharmacogenetic testing as a really single test. We should view it in the context of individualizing drug therapy, to my opinion, because we cannot sort out other sources of variability of risk of side effects like drug interactions or hepatic failure or so on. So the same conditions that we settle up for other conditions like hepatic failure in drug development, in pharmacovigilance, should also apply for pharmacogenetics. That's my first answer. And then I read out of this text that apparently the question is, should we have a bedside testing? This would mean that the patient perhaps could order even himself the testings. So that's a completely different field of discussion, whether or not pharmacogenetics should be something influencing health management or behavior of the patients or attitudes towards drug therapy. That's a completely different field, I think, to discuss, and probably too long to open this bottle here. Thanks.
The next panelist, please.
Yes, I think we had this perfectly in the session, the different fields of cancer, and which was given by Robert Diazio, and the topic given by Ron van Schaik, more concerned in the field of pharmacokinetics of some drugs not related to cancer, or the only example was the product tamoxifen. So we have to distinguish who is genotyping or making a genome analysis. In cancer, it's very clear this is a somatic disease based on mutations. So this is something where we have to go into the tumor tissue, and this is, to my point of view, is a field where molecular pathology is on demand, and with the advice also of human geneticists. So this field of cancer is clearly growing and rapidly growing, and it is in the hand of pathologists. All other fields who have something to do directly with drugs, with metabolism, with side effects. Of course, there must be an advice from clinical pharmacology, or at least from people who know what happens. If we go to the example of tamoxifen, it's very clear. It's a prodrug which does not act without metabolism by CYP2D6. So it's not a question of an association study. It's a question of knowledge if the drug is activated or not. And for Magnus Ingemann-Sundberg pointed it out also for clopidogrel. If you don't look to the right indication of the drug, of course, some association studies fail because clopidogrel was not usable. So that's why it's necessary to know what drugs are doing. And it's the same or similar level like TDM and psychiatry. You have to know about the action of the drug, what kind of drug are you using, and has the patients took the drug or not. From my point of view, there TDM is currently necessary before genotyping because you have to know about the behavior of the patient. That's something different. In this field, we have to know much more about the outcome of the study. This is still absolutely missing in the field of psychiatry, the benefit of the use of pharmacogenetics. What does not mean that pharmacogenetics is not useful, but we have not the evidence so far. So from my point of view, who should make it? It could be done, of course, in the hospital laboratory, but the data must be checked and together to give an advice to the clinicians. So just to have a bedside teaching perhaps would be a step which would be not so useful. We have to have the clear quality of the genotyping assay, and we have to know what are the consequences of this result of this test. And that is, I think, our task to make it and to bring up these full consequences of genotyping. And at the end of the day, of course, we have to make the legal issues. In Germany, it's currently absolutely a nightmare to perform pharmacogenetics. It's a question of compensation by the insurances, but also who is allowed to do it. Currently, we have to, in most cases, to use the advice of human geneticists, and this is absolutely not useful because they are used to treat hereditary diseases but not to give any advice to pharmacotherapy. Thank you very much.
Nick Mead, can I ask you to conclude the panel? Then we all desperately need coffee.
Thanks, David. I'll try and be quick. I just want to point out a difference between one of the first presentations we had yesterday by Federico to most of the other presentations we've had. And I think the point I want to make is that, Federico, you didn't really focus so much on the problems with getting testing done correctly and the issues around there that we've seen in a lot of the other presentations today. And I think that's the reason here is that you were speaking about pharmacogenetics for genetic patients in genetic disease. And this is quite a different issue, I think. So to answer the question here from my perspective, representing genetic patients, we've already done the testing. We know what our genotypes are already. So when the drug we were talking about yesterday, ivacaftor, came onto the scene, patients already know whether they've got the G551D mutation or not, and they can make the noise in advance of the drug even getting its market authorization. So I just want to underline something that I think probably we all know, but the distinction hasn't really been made clearly over the past 2 days, that there is quite a big difference here. Genetic biomarkers in genetic disease are already well understood and already tested for. These are genotypes which aren't being examined during the clinical trial. The therapy is being looked at in advance with knowledge of the genotype that we're looking at here. So we should maybe consider this fact, and I think that consider the different regulatory problems that these types of genotypes present. We don't need to validate the biomarkers because they're already well understood. So yeah, I just wanted to underline that point. But to actually answer the question here, from our experience of understanding genetic testing, when you test a patient, it's very important to be very clear about what test you're actually giving them and what the implications of the test is before you give the test, because a genetic test doesn't just apply to a single person but to generations that follow also. Thank you.
Thank you, Nick. So we have very interesting perspectives, very different perspectives from many sides on the table. My question actually goes to the agency. How do you want to continue this discussion? In which format and when? How is it going to happen? Marisa, can I ask you to comment?
Thank you very much. I think I will ask for confirmation, ask Georg about this vision, but what I got from the discussions today is that there is quite a spectrum of potential use of genomic knowledge. In some areas we will be more advanced, in other areas we are advancing very, very rapidly, for example in the cancer area, and I keep being convinced that pharmacogenomics is not an incremental science. And from what I understood in terms of impact on patients and public health, pharmacogenomics is incremental when it comes to learning and researching and accumulating evidence from this purely scientific point of view. But when it comes to the implementation in clinical use of such a science, my conviction is that is transformative in the regulatory sense, and we had a number of ideas which have been going around today and yesterday, how now this has impacted already the regulatory science, for example. How we moved from products to biomarkers. I mean, this is a tremendous transformation in the way the regulatory authorities have been looking at the science underpinning the drugs. Because tradition was always, the product. But in the FDA, at the agency, we change that and we look at what are the tools to get those products right. And this has been big transformation in 2008, so very recent. But I see also that in addition to the transformation in regulatory approaches, we heard from Professor Diasio the transformation in therapeutic regimens based on genomics, moving from surgery to treatment and then back to surgery. I was absolutely fascinated by the fact that maybe the way we look at the potential intervention is not just in regulating dose and selecting or deselecting patients, but because of the knowledge, being able to revisit entirely the way we approach treatment. And then I understand that the people on the front, like our friend in Netherlands, is really identifying the key bolts. What blocks this to become, regular things? And the answer I get from all the discussions, always one, also from the intervention from the public, is that people think at genomics in isolation, whereas still the patient is the patient with his whole phenotype, with his whole clinical history, his environment, and his habits. So I think as an agency, I would suggest to my management that we keep our interest in pharmacogenomics and try to embed it in the best possible way in the work of our committees. We have the chairman of our CHMP, decision-making committee when it comes to science and drugs. And then maybe we learn without reinventing the wheel what our colleagues in the FDA have been doing for information. And we know that there's been a big work done, and Michael Pakanowski has been very much involved in that, in rehearsing the information of medicinal products when it comes to pharmacogenetics. It's been done a lot of work, and we are also the agency in the process of doing the same work, but we are a little bit behind, so we'll have to hurry up. And then I think there are additional reflections which are carried jointly with the FDA, with PMDA, and many other authorities, how now we can reformulate the understanding of what a drug can do and cannot do because of pharmacogenetics. And I think there is a big challenge which is exemplified in the questions which were put across in the room in the post-authorization genomic research. How do we do it? How we organize ourselves? Because if it's true that we will also revisit the paradigm of learning, licensing, and learning again, then I think we have to take this on board. I don't know whether Hans-Georg, you want to add in any elements. So this is the way the agency would like to continue with all of you and identify how we can help for addressing the questions you as stakeholders may have at European and international level.